Dear students, now we are going to the third week of human embryogenesis, to the late gastrulation. So you already know that there are early and late gastrulation. Early gastrulation is going on during second week of human embryogenesis, and it includes delamination, which results in the formation of ectoderm and endoderm. So there is a bilayer embryo or embryonic disc, which is a result of early gastrulation. Now we are going to the late gastrulation. It includes formation of mesoderm, its main result of late gastrulation, and also it includes formation of axial complex and body of embryo. So processes of the late gastrulation are migration, invagination, also neurulation, formation of neural tube, somatization of mesoderm, formation of somites, and also formation of body of embryo. And result of late gastrulation is formation of three-layered embryo with an axial complex and formed body. So let's look what will be going on during third week of embryogenesis. So here we can see embryonic disc. It includes ectoderm and endoderm. It's viewed from the upper side of the embryo, so it's amniotic vesicle. Top of the amniotic vesicle was removed, and here we can see bottom of the amniotic vesicle, which is embryonic ectoderm. Here we can see embryonic ectoderm. It's anterior and posterior end of the embryonic disc. And migration begins in the ectoderm. So what is going on? Let's look at embryonic disc. Here we can see that it has oval shape, it's ectoderm, it's midline of embryonic disc, and some cells in the ectoderm, they begin their migration from anterior to posterior end of embryonic disc. They meet posterior end of embryonic disc and they turn back to the anterior side of the embryo. So they migrate to anterior side. But here, in this part of the embryo, there are some cells which are still migrating from anterior to posterior end of the embryo. Here we can see them. So some cells have turned back, but some cells are still migrating from anterior to posterior end. And what will happen here? Here cells will be accumulated, they will meet each other and they will form nodule. It's primitive nodule, it's group of cells which is result of cell migration. And also here we can see two rows, two directions of cells migration. They form dense spots, dense region near the midline, which is called primary or primitive streak. So primary or primitive nodule and primary or primitive streak. Here we can see those structures which are a result of migration, nodule and streak. And here we can see ectoderm, and in the ectoderm we can see formation of primitive streak. So result of migration in the ectoderm is formation of primitive or primary nodule and formation of primitive or primary streak. And here we can see primitive streak or primary streak and nodule. Here we can see them. It's ectoderm of the embryo and its streak and nodule. And look here, this streak, it has two dense spots on two sides and also it has groove inside. It's primary or primitive groove inside the streak. It's located on the midline of the embryo. And here we can see cross-section of this region of the embryo. It's ectoderm, it's endoderm, and somewhere here will be mesoderm developed. So it's ectoderm and it's groove, primitive groove, and two parts, two sides of the streak of primitive or primary streak. And some cells from the streak, they haven't enough place for their development, so they will migrate between the ectoderm and endoderm in the gap between two embryonic layers and they will form mesoderm. So let's look how it will happen. Here we can see microphotograph of this streak. It's ectoderm, it's endoderm, it's groove, and cells, they are falling in this region and they fill the space between ectoderm and endoderm. Here we can see ectoderm and endoderm, and cells inside the groove, they fall in this space and they form mesoderm. Here we can see ectoderm, endoderm, and cells from ectoderm, they fill this space and they form mesoderm. It's how mesoderm is developed. So ectoderm, endoderm, and cells, they fall between, and ectodermal cells, they form mesoderm, and mesodermal cells will produce intercellular matrix and it will form mesodermal layer, third embryonic layer. And here we can see ectodermal cells. After the migration, they begin their invagination. So it's called invagination, formation of mesoderm, which fills the space between ectoderm and endoderm. Cells migrate and fill the space. And also here we can see primitive nodule and streak and its groove. And what will happen to this structure? Why do we have this nodule? This nodule has pit inside, and this pit becomes deeper, and cells from nodule as well as cells from streak, they fall between the ectoderm and endoderm, but they form processes between ectoderm and endoderm. This process is called notochord. It's notochordal process growth. So it's located between ectoderm and endoderm, and its first axial organ, which is called notochord. I think you know that some animals they have cord or notochord, and human embryo also has notochordal process. It's located between ectoderm and endoderm, and it begins its growth from primitive nodule, from primary or primitive nodule, and it grows between ectoderm and endoderm. Here we can see ectoderm, endoderm, it's mesoderm which fills the space between ectoderm and endoderm, and it's notochord. And here you can see how notochord grows from the nodule. It grows between ectoderm and endoderm. Here you can see ectoderm, endoderm, and it's growing notochord. So here we can see first result of late gastrulation, formation of mesoderm. We already have mesoderm, and also we have completed invagination, migration and invagination, and result of them is formation of mesoderm together with notochordal process. And next stages will be neurulation and somatization of mesoderm, which will form axial complex of the embryo. Let's look what will happen. So here we can see ectoderm, endoderm, between them there is mesoderm and notochord. And notochord, here we can see notochord, ectoderm, endoderm, notochord, and also there are some in other structures, some organs which are called axial organs. They are notochord, and it's the first axial organ. And what is going on next? Notochord is located under the ectoderm, and it causes some changes in the ectoderm which covers this notochord, and it inducts beginning of the neurulation, special process of the formation of the neural tube. So ectoderm which covers notochord undergoes some changes which lead to formation of the neural tube. Here we can see ectoderm, notochord, mesoderm, and endoderm. And here we can see that ectoderm is completely flat on this stage, and later it forms neural plate, special region which covers notochord. It's called neural plate. And neural plate goes deeper. It forms neural groove, special neural groove. It goes deeper. Here we can see neural groove, and later it will be enclosed and it will form tube, neural tube, which will completely separated from the ectoderm. So here we can see result of neurulation. Here we can see completely separated skin or dermal ectoderm which covers the embryo, and its neural tube which has been separated from the ectoderm. So neurulation is a formation of the neural plate, and also neural plate will form neural groove, which will be closed and will form neural tube. And look here, it's embryo, it's upper side of the embryo, and here we can see neural plate and neural groove. And here we can see how it's enclosed and it forms neural tube. Here we can see that neural tube is enclosed beginning from the middle part, and also later, neural tube will be completely closed, and there are some openings on the anterior and posterior sides of the embryo. We call them neuropores, openings of the neural tube. Neural tube, here we can see it, it's dermal ectoderm and it's neural tube. And this neural tube will form a nervous system. It will form brain and spinal cord. So it's essential structure of the embryo. So ectoderm on this stage is divided into dermal or skin ectoderm, which will form coverings of the embryo. It will form epidermis of skin, and also ectoderm includes neuroectoderm, which will form nervous system. So we have notochord, we have neural tube, second axial organ, and third axial organ, it's somites. There are somites which are located on both sides of neural tube. Here we can see somites. So next stage of the development of the axial organs, it's mesoderm somatization. Here we can see ectoderm, endoderm, notochord, also there is neural tube, and undifferentiated mesoderm on both sides of neural tube. And neural tube causes some changes in the neighboring tissues which surround the neural tube. And this paraxial mesoderm undergoes changes. It forms somites. It undergoes somatization. So this paraxial mesoderm undergoes segmentation. It's divided into small particles, groups of cells, which are called somites. So what is it? Here we can see neural groove. It will be neural tube, and on both sides of neural tube, cell mass of the paraxial mesoderm is divided into groups, which are somites. Here we can see neural tube and enclosing of the neural tube. And on both sides of neural tube we can see segments of something. Those segments are somites. Here we can see somites on both sides of the neural tube. So neural tube and somites on both sides of this tube. It's called mesoderm somatization. Here we can see neural tube, and there are somites, groups of somites. So segmented structure is still present in our body in adulthood. We have vertebra, we have rib, intercostal muscles, nerves. So it's derivates of segmented embryonic structure. Each somite forms this segment of our body. And somite means body. So what will develop from somites? Somites will form dermis of skin, its connective tissue of skin. They will form skeletal tissues such as cartilaginous and bony tissues. And also somites will form skeletal muscles which move our body. So third embryonic axial organ, it's somites, groups of somites. There are about 45 pairs of somites. So there are pairs of somites. One somite is on the right side, another on the left side of the embryo. And here we can see chicken embryo. It's neural tube. It's developing brain. And there are somites on two sides of the embryo. And also here we can see somatization. Some somites, here we can see 24 hours in the chicken embryo, and here we can see later stage. This embryo has more somites than this. And also here we can see somites. There are segments which are located on two sides near the neural tube. So there are following axial organs: notochord, neural tube, and somites. Please note that there are three axial organs of embryo. It's axial complex of embryo, and it plays essential role in the development of the embryo in the following stages of embryogenesis. So here we can see ectoderm, its endoderm, its notochord, neural tube, and somites. And here we can see ectoderm, endoderm, its notochord, neural tube, and somites. And also we can see that ectodermal structures are shown in blue. It's skin or dermal ectoderm, its neural tube or neuroectoderm. Endoderm is shown in yellow, and mesodermal structures are shown in different shades of pink. Here we can see somites, it's paraxial mesoderm. Also there is intermediate mesoderm on two sides of somites, it's called nephrotome. And also there is lateral mesoderm, it's called splanchnotom. Splanchnotom, it has two layers, parietal and visceral layer. It's parietal layer and visceral layer, and between there is a cavity which is called coelom, coelomic cavity. And also we can see here light pink shades, something fills the spaces between all those structures. It's mesenchyme, which is a part of mesoderm. It's embryonic connective tissue which fills the spaces between embryonic structures. So it's undifferentiated mesoderm or mesenchyme. And on this stage we can see that embryo has flat shape, mainly flat shape. It's embryonic disc, ectoderm, endoderm, and everything between this. And we know that we have three-dimensional body. So what will happen next? And next, there is next stage, which is called folding of the embryo. Embryo from the flat structure, from flat embryonic disc, will become to be three-dimensional structure. Here we can see before, embryo before the folding and after the folding. Here we can see that embryonic structures, they were closed in the three-dimensional structure. Here we can see ectoderm on one side and on another side, and they were fused to those layers. Also, endoderm was enclosed in the tube, endodermal tube. It will be intestinal tube which will form digestive tract. Also, splanchnotomes, layers of splanchnotomes, they were enclosed, and parietal layer was fused with parietal layer from another side, and also visceral layers also were fused. So here we can see three-dimensional body of embryo. And how is it provided? What is going on during flexing of the embryo? Let's go to the amniotic vesicle and yolk sac. Here we can see two vesicles, and place of their attachment it's embryonic disc, place where two vesicles were attached to each other. It's embryonic disc, and you know that only this part will form embryo. And here we have extraembryonic ectoderm and extraembryonic endoderm. This one is embryonic ectoderm and this one is embryonic endoderm. So on this stage, those vesicles are almost the same, but later amniotic vesicle grows faster than yolk sac. And here we can see that it grows faster and it surrounds yolk sac outside. And here we can see that embryonic disc undergoes flexing and it becomes to be concave. And later ectoderm still is growing faster than endoderm and surrounds it and forms body fold. And this body fold separates body of embryo from extra embryonic organs. So it will be body of embryo. Ectoderm now surrounds embryo outside. It will form coverings of the embryo. It will form epidermis of skin, outer layer of skin. Endoderm enters inside the embryo. It will form intestinal tube. It will be digestive system, digestive tube inside the embryo. Here we can see umbilical cord. It will be umbilicus and its umbilical cord. And extraembryonic ectoderm forms very big sac. It's amniotic sac. And now we can call this amnion. It's amnion. And you know that embryo develops in some cavity, in some sac. So embryo develops in the amniotic cavity, which forms this sac. And its yolk sac, its embryonic part, its embryonic endoderm, it's present inside the embryo, and its extraembryonic part of endoderm, it forms vitelline duct inside the umbilical cord and extraembryonic yolk sac, and in human embryogenesis it has rudimental structure, so later this sac will be reduced. And let's look at this diagram. Here we can see amniotic vesicle, its yolk sac yellow, and amniotic vesicle surrounds yolk sac outside. Here we can see and forms body fold, and embryonic body becomes to be three-dimensional. Here we can see result of body flexing. It's formation of body fold and formation of body of embryo. And here we can see cross section of the embryo. It's notochord, neural tube, somites, it's amniotic vesicle, it's yolk sac, and it forms intestinal tube which enters inside the embryo, and amniotic vesicle surrounds yolk sac outside. Here we can see this process. And here we can see result of body flexing. It's formation of three-dimensional body of embryo. So here we can see structure of embryo before the body flexing. It's ectoderm, it's neuroectoderm or neural tube, it's endoderm, it's notochord, and all other structures they are mesodermal structures. There are somites or paraxial mesoderm, intermediate mesoderm or nephrotomes, and lateral mesoderm or splanchnotomes, and coelom between them, and also there is mesenchyme which fills the spaces between all those structures. It's flat two-dimensional embryonic disc before the body flexing. And after we can see later stage of the embryonic development. Here we can see ectoderm, it's endoderm, it's body fold which separates body of embryo from extraembryonic organs. Endoderm forms intestinal tube on this stage. It's splanchnotom parietal layer and visceral layer and coelom between them. It's notochord. It's developing somite which forms dermatome, sclerotome and myotome. It's neural tube, and also we can see here nephrotomes. They form tubes which will form urinary system, and mesenchyme which fills the spaces between them. It forms blood vessels. Also there are developing cells inside blood vessels. Here we can see, and also here we can see blood vessels. And also mesenchyme will form connective tissues of embryo. So here we can see later stage of embryonic development. So here we can see body of embryo before body flexing and after body flexing. Here we can see result of this stage. So we can see here result of late gastrulation, formation of three-layered embryo which has ectoderm, endoderm and mesoderm, and also it has embryonic axial organs, and also body of embryo is formed, it's three-dimensional, so it's result of the late gastrulation. So in the next topic, in the next part of this video, we will talk about differentiation of embryonic layers. What will happen to all those layers? What will happen and what will be developed from ectoderm, endoderm and mesoderm? Thank you for your attention.